Is it
Be
recording?
sure to bring you a pencil next time and a Scantron. We'll have 50 multiple choice questions as usual.
Where would be a good place to put the recorder?
I'd say by this sink right here. That was y'all. I'm going to show y'all a lot of demos. Be over here most of the time. When you come in here next time, if I was you, I'd go to a station and write some things down from memory on the back of the answer sheet. I'll give y'all some time to do that. Some charts for blood type and transfusions, maybe. Some results charts, maybe. So you have some time to do that, whatever you want from memory, on the back of the answer sheet. We better start Lab 9 on the right-hand column. Now on these demos, I will pull questions off the cards. So be sure to stuff in the cards in your notes for questions coming up. So recognize this right here as an alcohol rub. On the skin, I'd say alcohol kills bacteria good. It also does de-germing. It removes bacteria really well. Recognize this as an iodine rub. On skin, iodine kills bacteria excellent on the skin. The best that we have. Recognize this as a vacutainer for drawing blood. Now, be sure to know that the gel separates the blood cells from the plasma when they spin these. What you can't see too well, but it's probably in there, is this clear stuff called heparin, which keeps the blood from clotting up. Now, recognize this as a Bactec system for a blood culture. This is for a blood culture. Does the patient have bacteria in the blood? Is a big question here. And I sometimes ask the recipe question. It's 1 part patient blood to about 10 parts nutrient broth. It'd be blood from the same patient in each bottle. Now, what I call the light green cap here is growing bacteria that are called aerobic. What I call the red cap grows bacteria called anaerobic. And after they incubate these for a while, they test inside the bottle for CO2 because all bacteria give off CO2. So a lot of CO2 in there means they got bacteria in their blood. They got a blood infection. This right here is called the SEPTI-CHECK system. SEPTI-CHECK. Also for a blood culture. Does the patient have bacteria in the blood? It's the same question again. This time around, the colors are different. Be careful. The red cap side is growing bacteria called aerobic, and green cap is anaerobic. Now take a look at the left-hand side, and it says a chart on the blood types. You may see a question like this next time. If some person is type O, what do they have for alloantigens? Nothing. If they're type O, what do they have for alloantibodies? They have A and B. So I'm going to pick out one of those 4 blood types and ask you what they have. So read the questions carefully. Does it say alloantigens or alloantibodies? Be careful. Now it says the chart or recipient donor. I'm not going to ask you this next thing when I mention this. We're trying to avoid in this chart right here matching up. In the same person, A antigen, A antibody. Can't do that. You can't match up the B antigen and the B antibody. On those questions that are practical right there, I'm going to say things like, if your patient's type A, what can you give them? O and A will be your answer. So I'm going to give you all one of those recipients. You've got to pick out for your answer next time the correct donor blood on that chart. The universal donor is type O. Why? On the donor side, you worry about the antigen. Type O has no antigen for blood typing. The universal recipient is type AB. On the recipient side, you worry about the antibodies. But type AB has no antibodies. We're going to mark out the next line there. It says ABO incompatibility, IgM. Let's mark that out. Now find your notes on the RH factor. RH factor. This protein on your red blood cells makes your blood type positive, like I'm A positive, for example. It is dominant to be positive, so 85% of all people are RH positive. Only 15% are RH negative. It talks about RH antibodies. Only RH negative people can form or make RH antibodies. If you're like me and you're RH positive, you can't make these. You don't want to. You'll be attacking your own self. It also says when, as in, when should an RH negative person be exposed to RH factor? Well, in a blood transfusion mistake, an RH negative person could be exposed to RH factor. Also, having a baby can expose a negative mother to positive cells from the baby. We're skipping on the handout that one little chart, although you have to know it. If your patient's RH positive, the donor can be either way, positive or negative, doesn't matter. If your patient is RH negative, the donor blood has to be negative. You don't want to expose them to RH factor because they tend to react to it. It's foreign to them. Be sure to know the big chart for blood transfusion. We have 8 possibilities. Again, I'm going to give you all the recipient, and you pick out the correct donor blood. You may see a question like this next time. If your patient is A negative, what can you give them? O negative and A negative would be the answer. So can you pick out the correct or safe donor blood? We also talked about the 2 main mistakes in a blood transfusion. In the AB mistake, you have matched up in the same patient, A antigen, A antibody. Or you match up in the same patient, B antigen and B antibody. If you make on some patient the AB mistake one time, they might die. In the RH mistake, you gave an RH negative patient RH positive blood. Here they will not die, but they might feel sick as they react to it. There will be some questions next time, like the ones that are on page 15. In your lab manual, look at page 15. I'm going to give you all next time some questions like, here's the recipient's blood type, here's the donor's blood type, what's your conclusion? It may be okay, it may be a B mistake, or RH mistake, or maybe both mistakes. Let's try those for practicing lab manual there. Number 1 says B negative patient, O negative donor. That's okay. Number 2, O positive patient, A positive donor. That's an AB mistake. They might die. 3 says AB positive patient, B negative donor. That's okay. 4, A negative patient, A positive donor. That's RH mistake. They might feel sick there. 5, A negative patient, B positive donor. That's both mistakes. They may die from the AB mistake. 6, AB negative patient, B positive donor. That's RH mistake. They might feel sick there. On your practical, not the exact same questions there, but they'll be similar. Kind of use your big chart and figure out the conclusion. Now, the review sheet says the true universal donor. If I say the word true, I'm going to add in the positive negative part. So O negative is the true universal donor. The same reasoning. On the donor side, you worry about the antigens. O negative has no antigens. The true universal recipient is AB positive. The recipient side, you worry about the antibodies. AB positive has no antibodies. I sometimes break that down for your answer though. Because they're type AB, they don't have A and B antibodies. Don't have those. Because they're RH positive, they cannot make RH antibodies. So we have no antibodies and no problem. Now the problem scenario for RH factor and having babies. To have a problem here, the mother has to be RH negative. Because if she's negative, she can make Rh antibodies. She can. That could be a problem. To have a problem here, the father has to be Rh positive. If he is, the couple can have positive babies. A negative mother and a positive baby, that's the problem. So again, if he's positive, they can have positive babies, that could be a problem. If that couple right there, negative mother, positive father, have two children that are both positive, and nothing is done about it, like in the old days, the second child could die before it's born. Because in the first birth process, the blood cells can mix, and mom's exposed to Rh positive cells from the baby. She reacts. She makes millions of Rh antibodies to attack that foreign shape, foreign to her. Now be sure to know this for next time. The antibodies called Rh antibodies are always IgG class. They can cross the placenta. So during her second pregnancy, the antibodies can cross and attack the red blood cells and destroy those from the second baby. That baby could die before it's born. We now use, to deal with this problem, RhoGAM. This is injections to the mother if she's Rh negative. Be sure to know this. RhoGAM prevents the mother from making Rh antibodies. If she doesn't make those antibodies, then there's no problem. The funny thing is, RhoGAM is made of Rh antibodies. They destroy red blood cells from the baby in mom's system before she can react to them. That's the idea. We're going to skip some stuff on serology in your notes, but find your notes on front type and back type, some of the details. On the blood typing tests, we're looking for a reaction to happen called agglutination. Agglutination means cell clumping. If on some card you have antibodies and red blood cells binding together, the clumping thing happens, we see grainy, obvious change to grainy. As a result, it's positive. So front type, blood type. Test a patient for 3 blood typing antigens. A antigen, B antigen, Rh factor. Now the right-hand column says diagram front type. So here's the card to look at. The front has the diagrams, the back has the answers. On your practical, I'm going to draw these one at a time on a card. I'll tell you this is a front type. What's the correct blood type? So use this card to kind of quiz yourself on what the answer would be. That's kind of what I call the easy kind, where you think forwards. Not so easy is the back type. The back type is where you test the patient for blood typing antibodies, as in A antibody and B antibody. So here on the back type card, you kind of think backwards. Who has the A antibody? That's type B, for example. So use the card, the back has the answers, kind of quiz yourself. And the question will say, this is a back type diagram, what's their blood type? Okay, lab 10. Lab 10. Now a lot of information from lab 10 notes has been left off. That's a good thing. But it does say on the handout, food poisoning. Now, number 1 cause of this, in our country most common for sure, is called salmonella. Number 2 is called Campylobacter, number 2. The so-called raw chicken bacteria are 1 and 2. It also says to know a gram-negative raw that causes gastroenteritis. That's salmonella. Know a gram-negative rod that causes bacillary dysentery. That's called Shigella. Shigella. We tried a couple of lab tests for gram-negative rods to identify. You may see a question like this next time. What makes MacConkey agar selective? The answer is crystal violet and bile salts. Go with selective. They mess up peptidoglycan. They therefore kill gram-positive bacteria. They don't kill gram-negatives. On MacConkey agar, only gram-negative bacteria can grow. What makes MacConkey agar differential? It's the lactose and neutral red. Now, sometimes I ask this question. In MacConkey agar, what's the pH indicator called? That's neutral red. So the pH starts out basic, Neutral red starts out clear. If your bacterium breaks down lactose sugar to make acid, the neutral red changes to a pink or purple type color. It's supposed to be pink. So the right-hand side says McConkey-Auger results. On the practical, I'm going to say this is a McConkey-Auger plate. I'll tell you that much. What's your conclusion? Well, first of all, we have growth. Must be gram-negative. It grew. We see a color change to pink-purple type thing, so they can break down lactose. They can break down lactose. In our case, this could be E. coli. In our case, it was E. coli. Here we have growth on the plate. Must be gram-negative, but no color change. I call these clear colonies. So the bacteria cannot break down lactose. They cannot. In our case, we had two possibilities here: Salmonella enteritidis or Pseudomonas aeruginosa. Those two cannot break down lactose. Now a more detailed test for gram-negative rods called the TSI slant. TSI. In this case, the pH indicator is called phenol red. If bacteria break down sugar to make acid, the phenol red changes from red to yellow. In that test tube, be sure you know how to read the 4 test results. If the bacteria can break down glucose, the bottom or the butt of the tube turns yellow. If they can break down lactose and sucrose, the slant on top turns yellow. If they produce lots of gas called CO2 and H2, you'll see bubbles or gas pockets. If they produce H2S gas, you see a black color. Now, know the chart we had on 3 possibilities. E. coli was like plus, 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 minus. Salmonella enteritidis was plus, minus, plus, plus. Pseudomonas aeruginosa is all the way minus across the board. So, on your practical, these 3 tubes have be the same station. So I think in these terms. The yellow one is E. coli. If you see some black color, that's Salmonella enteritidis. The red one is Pseudomonas aeruginosa. But you may see a question like this. Can this one here break down glucose? Yes. Lactose, sucrose? Yes. Gas, I see a gas pocket, things pushed apart in there. Gas is positive. H2S, no. So kind of jot that chart down, maybe keep things straight. This one here, you can see these 4 results pretty well. The butt looks pretty yellow, glucose is positive. The top part of the slant, way up here, is still red. Lactose sucrose is negative. We do see some bubbles in the front, and we see some black color as well. So I see those pretty well in this particular tube right here. It didn't react too much. Sometimes they do. And of course, Pseudomonas here is just all the way negative across the board. So the same station they'll be at. By the way, the numbers on the tubes and plates next time will be different numbers. Be the question number next time. Question number. Okay, Lab 11. In Lab 11, thank goodness, a lot of things are left off. We're going to skip gonorrhea and meningitis and syphilis. We'll ask questions on those in lecture in the final. But find your notes for now on tuberculosis. Let's skip to tuberculosis. This is such a big problem. I want to talk about this in lab for sure. I want you to recognize this demo over here as being a Sage sputum container, like a brand name, Sage sputum. Use this to collect sputum when the patient does not have artificial tracheal tube. They can cough it up in their lungs, put it in here. This right here you should recognize as a Lukey tube. Lukey tube. To collect sputum when the patient does have artificial tracheal tube. You got a vacuum suction the sputum out of their lungs. Now the left-hand column says the acid fast stain. This is used primarily to identify the mycobacteria, which includes mainly TB. The first step here is called carbofusion. This stains all the bacteria red. After step 1, we have mycobacteria like TB, red. The non-mycobacteria like staph, pneumonia, also red. Now be sure to know this for next time. Carbofusion is lipid soluble. the red dye dissolves right through lipid. The TB bacterium has a strained cell wall with a bunch of lipid. So the red dye penetrates through that, and they stain red inside and out. The non-mycobacteria like staph are barely stained red. Step 2 is called acid alcohol. This washes the carbofusion out of the non-mycobacteria. After step 2, we have mycobacteria red and non-mycobacteria clear. Step 3 is called methylene blue. This stains the non-mycobacteria blue. So your final results are mycobacteria red and non-mycobacteria blue. You may see a question like this next time. If you take sputum, do the acid fast stain, and they wind up stained blue, what do you think? Pneumonia. If they stain red from sputum, that's tuberculosis. Now it mentions there a faster and more mass-produced test for TB, the 7H12 Middlebrooks media test. This is used for tuberculosis. This liquid media contains a bunch of carbon-14, which most bacteria would tend to ignore if they're around it, but only TB bacteria take it in and use it. They make CO2 from it, which they give off eventually. So mix together in a big tube your sputum from the patient and this medium, and after a while of incubating, test for CO2 in the tube. In this case, CO2 means it's tuberculosis. Now skip to your notes on HIV testing. How do you test someone for HIV? The first test they do, the main test they do, is called indirect ELISA. This tests the patient's serum for HIV antibodies. Not the virus, HIV antibodies. If they've made those antibodies, then they have HIV. Remember, in the ELISA test, the E stands for enzyme. That creates for us a color change for the result. If that test comes back positive, then do a second test to verify. It's called the Western blot test. This tests the patient's blood for HIV proteins, or epitopes. If they both come back positive, the patient's then told they're HIV positive. Now skip to your notes on Lab 12. Let's start Lab 12 in the right-hand column with some demos. This right here is aerobic culturette. Aerobic. There's a cotton swab in there. Know your examples. Use this for a throat swab, a nasal specimen, abscess, or genital specimen. This is anaerobic culturette. Anaerobic. The same four examples: throat, nasal, abscess, and genital. We're going to skip the bag and the disc down there, but know what it is and the four examples. We'll use it for you. This is called a wire culturette. It's a very small cotton swab in there for urethral and nasopharyngeal specimens. The left-hand column now mentions hemolysis. This is the bursting of red blood cells. Now, on the lab practical, I'll have these three plates here at the same station. You got to figure out which one's which and what's the best guess in each case. Now, these are reacted enough that the plates look really good. So take this one here and hold it up, and where the bacteria are, it's definitely lighter where they are. But you can't see through it, it's not clear. This is called alpha hemolysis. And your best guess is called Streptococcus pneumoniae. This one here, when you hold it up, you can see right through it. See my finger right through it. This is called beta hemolysis. It's clear. You can see through it. If you see beta hemolysis by a strep, it's probably one called strep pyogenes, strep throat. Hold this one up, and where the bacteria are, it's still blood red. It's not lighter. It's not clear. This strep is non-hemolytic, and it's probably one called Enterococcus. Now, by the way, the card says that it's light red, just to help you out, but on the practical, it won't say that. It'll say, this is a plate of hemolysis, what's your conclusion? You got to know it is alpha, and probably one called strep pneumoniae. So those three plates at the same station, Gonna pick them up, compare which one's which. Now the left-hand column says Streptococcus on a gram stain. I tend to ask these in words and individually. If I say Strep pyogenes or Enterococcus, it's a gram-positive coccus in chains. Strep means chain. I could ask the exception though. Strep pneumoniae is a gram-positive coccus in pairs. The left-hand column now has details on Strep pneumoniae. This does alpha hemolysis. To find out for sure if it's this one, the lab does an Opticon test. Connect pneumoniae with Opticon. The drug called Opticon can kill only one kind of strep, that's pneumoniae. So I'll say, this is an Opticon test. What's your conclusion? Well, you got to pick it up and look at it, either this way or the other way, or hold it up to the light. But the idea is this. The clear zone is red because around that disc, they were killed. They couldn't do any hemolysis. Away from the disc, they're doing hemolysis and changing things. So the red zone is the clear zone. This is Strep pneumoniae. They were killed. Here we have no red zone. We have no clear zone. It's just not pneumoniae. So all that matters here is pneumoniae, yes or no. The clinical significance for this one is three things mainly. Strep pneumoniae can cause pneumonia, meningitis, and middle ear infections. Strep pyogenes does beta hemolysis. The so-called culture test for this one is the bacitracin test. Again, bacitracin kills only one kind of strep. That's pyogenes. I'll say this is a bacitracin test. What do you think? Well, once again, the clear zone right here is red. So away from that disc, they survived and are doing hemolysis. But around that disc, they're dead. They did nothing, so it stayed red. So again, the red zone is the clear zone. This is Strep pyogenes. This is strep throat. Here we have no red zone, no clear zone. It's just not pyogenes. All that matters here is pyogenes, yes or no.
So you will tell us what test it is?
Oh, absolutely.
Okay.
You bet. This is a bacitracin test. What do you think? Now, this right here is sometimes called the quick test for Strep pyogenes or strep throat. Make a note for your card. It's called a direct ELISA test. Direct ELISA. There's an enzyme in there for a color band to appear. This is a direct ELISA test for Strep pyogenes or strep throat. Results are back in about 10 minutes. Much faster. The clinical significance for this one, I'll mention three things that sometimes happen in progression. Stage 1, strep throat. Stage 2 is scarlet fever. Stage 3 is rheumatic fever. Now some more details on Enterococcus. This is a non-hemolytic strep. The test is called bioesculin. Here is a tube of bioesculin agar. Only one kind of strep can survive the bile and grow on here. That's Enterococcus. As it grows on here, it breaks down esculin and makes iron sulfide, which is black. So I'll say this is a bioesculin test. What do you think? Well, this right here is definitely Enterococcus. It survived the bile. This is Enterococcus. This one here is definitely not Enterococcus. It's not. And whatever it is right here could not handle bile. Bile killed it. The clinical significance for Enterococcus is two things mainly. Enterococcus can cause UTI and bacterial endocarditis. Now we'll switch over and talk about staph. Mannitol salt agar is used to grow identify staph. Your test question might say this: what makes mannitol salt agar selective? It's the salt. Most bacteria cannot survive that much salt. They can't grow on that plate. The main exception to that would be Staphylococcus. Because staph hangs out in clusters, the cluster effect, staph survives the salt and grows on there. What makes mannitol salt agar differential is mannitol sugar and phenol red. If bacteria can break down mannitol sugar, they're going to produce acid. That's going to make phenol red change from red in yellow. So the right-hand column says mannitol salt results. I'll say this is a mannitol salt plate. What's your conclusion? Here we have three conclusions. They grew on there. Must be staph. They grew. They did not turn yellow. The bacteria cannot break down mannitol sugar. They cannot. And it must be the one called Staph epidermidis. It's that one. It doesn't turn yellow on here, but it did grow. It's Staph epidermidis. Here we also have three conclusions. Once again, it grew on here. Must be staph. It did turn yellow. The bacteria can break down mannitol sugar. And in our case, we have two possibilities we're thinking about. Could be Staph aureus or Staph saprophyticus. We're going to think about, for our answers next time, just the three main types of staph. There are some more. We're going to focus on those three. This here could be Staph aureus or saprophyticus, either one. The left-hand column says Staphylococcus on a gram stain. I tend to ask these in words and individually, but they're all the same thing. And if I ask any of those, it's going to be a gram-positive coccus in clusters. Staph means clusters. Now some more details about Staphylococcus epidermidis. The only test I know of is mannitol salt. Because again, if it grows on this plate and does not turn yellow, then you're done. It is Staph epidermidis. It's that one. The clinical significance here could cause a UTI or a blood infection. Staph aureus makes mannitol salt turn yellow. The traditional test here is called the coagulase test. Coagulase enzyme makes plasma clot. But only one kind of staph makes coagulase enzyme, and that's Staph aureus. So on your practical, they'll be sitting there, I'll say, this is a coagulase test, what do you think? You got to pick them up. You got to tilt it. If it sits there in the bottom as a clot, that's Staph aureus, coagulase positive. It's still very watery, not clotted, it's not Staph aureus. Could be epidermidis or saprophyticus, but this is not Staph aureus. The clinical significance for Staph aureus, I mentioned 5 things. It's the main cause of pimples and boils. It can also cause food poisoning, pneumonia, and blood infections. Staph saprophyticus makes mannitol salt turn yellow. The test here is called novabiacin. The concept is kind of backwards. Novabiacin kills all types of staph except saprophyticus. Kind of backwards. I'll say this is a novabiacin test. What do you think? Well, here I see a clear zone the size of a quarter. So this is not saprophyticus. I see a clear zone. It's not. Could be aureus, epidermidis, not saprophyticus. Right here, we definitely have no clear zone at all. No clear zone. So this is saprophyticus. The clinical significance for saprophyticus is mainly UTI in females. Now, I have 9:05. We have some class time for a while now. Look at demos. On the board, it says today from 10:30 to 11. We can look at demos some more if you like. Also 12 to 2 today. Tomorrow, I have lab practicals in here all day, but I can do demos from 4 to 5 tomorrow. So I'll be in here in my office. That's all.
I didn't take this, didn't I?